Hello, my name is Christopher Tam, and this is the Network2Canvas tutorial. Network2Canvas is a web application that provides an alternative way to view networks. Though networks are central in computational systems biology research, visualizing them can be a challenge. For networks larger than 100 nodes and 200 links, ball and stick diagrams fail to convey much information. To address this, we developed Network2Canvas, which provides this alternative way to view networks. The header of the interface allows you to choose between drug canvases or the gene canvases. The default is the gene canvases. The third option in orange leads to the Canvas node learning game, which will be discussed in a different tutorial. And finally, you have the help link in the corner, which links you to the written tutorial, as well as the GMT files and the associated scripts used to create these canvases. The main part of the Network2Canvas user interface is the Canvas object. The Canvas object has a zoom-in feature which is accessed through the mouse wheel. This object also supports panning by holding down the mouse button and moving side to side. When your mouse pointer hovers over a node, the name of the node will be shown next to the pointer. Clicking on a node will bring up a list of genes associated with that node underneath the Info tab. The drop-down box allows you to choose between various canvases. All the canvases are in different categories: transcription, pathways, ontologies, disease and drugs, cell types, and miscellaneous. Underneath the canvas object, you can select between three different views: the main view, which is the current object, and the network and p-value view, which are only enabled when you run enrichment analysis. If you click on the Canvas Options tab, you are able to change the properties of the canvas object. One property you can change is the color. Another property you can change is also the color scaling, which will allow you to see better contrast between similarly colored nodes. By default, the color scale is to 0.25. If you select Reset Scale, the canvas object will revert to its natural color weighting. The Center Canvas button is used after you zoomed in or panned, and it will reset the canvas to its natural zoom-in level. Another feature of Network2Canvas is the find by term or element function. Specific terms in the gene set library of the canvas can be found using the find by term function. After submitting a list of terms, we will use bile acid biosynthesis, for example, and submitting it, it will indicate where it is on the canvas. Additionally, the find by element function has an option to search gene set library terms by submitting genes that are associated with it. So if we use ADHFE1 and use find by element, this will indicate all the terms that have ADHFE1 inside their gene list. Enrichment analysis is another feature of Network2Canvas and can be performed after clicking on the Analysis tab. By adding a list of genes and choosing your color of your indicated marks, so our default will be white, Network2Canvas runs enrichment analysis using the GMT file associated with the canvas. So in our case, we will be using KEGG pathways as our GMT file. So we'll click Submit for our gene list, and what's outputted is the top 20 most significant terms underneath our Analysis Output tab. These p-values are computed using the Fisher exact test. So right now on the canvas, our brightest terms correspond to the most significant terms in the analysis output, sulfur metabolism being the brightest. Going back to our main view has the most significant terms indicated using a circle, the most significant being p-values under 0.05. As you can see, we only have 4 terms that meet this criteria. The more opaque a circle is on this canvas, the more significant it is. So as you can see, sulfur metabolism is the most opaque. The network view takes these 4 enriched terms and then tries to connect them using their nearest neighbor distance. Also, you can see the degree of clustering in your terms by looking at the clustering z-score. The more negative your z-score is, the more clustered it is. As you can see here, our terms aren't really that clustered. You can also click on this link to download your p-value table with overlapping genes. Finally, the last feature to show you is the Download Canvas as a Networks link. This allows you to download the current view. All you have to do is right-click, Save As, and then download it as SVG. The good thing about SVG images versus raster images is that no matter how big you make them or expand them, they never get pixelated. You can open up this SVG image in Adobe Illustrator or Inkscape. So that's it for the Network2Canvas tutorial.